Okay, now we're going to take a look at RNA synthesis. It says in transcription, remember transcription is that first step toward making a protein. And that's where you go from the DNA to the messenger RNA. So in transcription, RNA polymerase separates the two DNA strands. Now I'm going to highlight RNA polymerase. That's going to be important to know right there. So if you can highlight, go ahead and highlight that. So RNA polymerase separates the two DNA strands. RNA then uses one strand as a template to make a complementary strand of RNA. RNA contains the nucleotide uracil instead of the nucleotide thymine. Okay, when we were doing the DNA replication, we said that A's go with T's. Well, there are no T's in RNA. So in RNA, there's going to be a U instead. Uracil is the base that takes the place of thymine in RNA. Okay, so we're going to label the RNA and label the DNA. And then label the missing nucleotides. Okay, so what is double-stranded would be the DNA. So this right here, we have both sides of the ladders. Both sides of the ladder there. With the rungs still connected. So this would be our DNA. DNA is a double helix. Two-stranded molecule. It's like a twisted ladder. Okay, so the circle represents RNA polymerase, opening it up and laying down the new bases. Up here we have the single strand that's coming out. This is being matched up. The RNA polymerase is working right here. It's going in. Polymerase, remember, is going to put in and proofread. So it's going to put in the complementary bases here. And it's going to proofread to make sure they're in the right place. Polymerase puts in and proofreads. So this is coming off here with just the one set of bases. It's not paired up anymore. Single strand, one set of those bases. This will be the RNA. And right here under the RNA polymerase, I'm going to go ahead and put us a little note so we remember that puts in and proofreads. Polymerase is going to put in and proofread. The P's go together. So puts in and proofreads. Important to know what polymerase is doing. Okay, right here we're going to match our bases. If I have a C here, C is always, doesn't matter if it's DNA or RNA, C is always going to go with G. So that's going to be a G right there. And right here we have, whoops, I wrote that on the wrong one, didn't I? It's supposed to be C, that's the one going to the C, it's right there. G-A-N-I-N-E, guanine. This is an A. A, when we were making DNA, we said A goes with T, but this time we're making an RNA strand, and there are no T's in RNA. So this is going to go with a U, uracil. Uracil takes the place of thymine when we're talking about RNAs. So an RNA, A goes with U. And this is a G. G still goes with C. This guanine is going to go with the cytosine. Here we have a T. T's are still going to go with A. So if we have a T on our DNA, it's still going to make an A on the RNA. This is going to go with adenosine. What's that part? Single-stranded RNA coming off. RNA is just going to be a little section of the DNA at a time, and then it's going to copy that message so it can take that message out of the nucleus, out to the ribosomes. And the DNA cannot leave the nucleus. So the RNA, that's what's called mRNA, messenger RNA, is that messenger that's going to carry those instructions for making those proteins. It's going to carry it out of the nucleus through the little pores in the nuclear membrane. It's going to carry that message out of the nucleus, out to the ribosomes where the proteins are being made. So that's the instructions for making proteins, but it has to go to the ribosomes because they're the ones that are doing the making. And, use the questions, answer the questions, you circle the correct answer. 4 says, in DNA, blank is always paired with cytosine. Well, cytosine starts with a C, so it's going to go with guanine with the G. Cytosine and guanine go together. In RNA, blank replaces thymine. There's no thymine in RNA. It's replaced by the U-uracil. Number 6, in DNA, blank is paired with adenine. So this says DNA. In DNA, A goes with T. So in DNA, we still have thymine. What's that? In DNA replication, a cell copies its DNA. Both strands of the double helix are used as templates to make complementary or matching strands of DNA. In DNA transcription, a single strand of DNA is used as a template to generate a strand of mRNA. So one side can code for one protein, and another part can code for another protein. The other side can code for another protein. So that single strand of DNA can code for many, many proteins. So if we're going for complementary DNA, we'll go backwards to what our original template is, and then our template is also what we're going to use to make our messenger RNA. So before we can go to the messenger RNA, they did an example here. If we're going to DNA, the A's are with T's, notice. But if we're going to RNA, then that T would not go with an A, it would go with a U. No T's in RNA. So if you think you should, if you look at it right here, you think you should be putting a T. I should go over the template over here. If you're looking right here, you think you should be putting a T with that A. Well, if it's an RNA, you're going to put a U. Okay, complementary DNA, we're going backwards. We're going to our template for our DNA. We have a G, so a G would go to a C. Another G would be a C. And a C would be a G. G would be a C, G would be a C, C would be a G. So there's our template, and that DNA template is what we're going to use to make our RNA. We're going to be looking at that. Our C, we have C and C, that would be GG over here. That G would be a C, C would be a G, C would be a G, and G would be a C. And then we can remember our triplets, our triplet codons. When we have GGC would be a triplet, that would code for an amino acid, and then GGC would code for that amino acid again. Notice that our messenger RNA ended up the same as our complementary strand of DNA because we didn't have to worry about T's and U's. Okay, so for our messenger RNA, we're going to go all the way back into our template. From that messenger RNA, we're just going in reverse. So if it coded for an A, it had to have been a T in the DNA. If it coded for the C, then it was a G. If it coded for this G, it was a C. If it coded for this U, it was an A. If it coded for this A, it was a T in the DNA. And if it coded for this G, it was a C. That's our template. Then we can use our template to, if we're going to do DNA replication, then it would code for a complementary strand of DNA. In which case the T would go to an A. That G would go to a C. C would go to a G. Here are A. This is complementary DNA, so A is going to go to a T. That T will be an A. And that C will be a G. And the messenger RNA that we have there looks very similar to this, except it has a U in the place of the T. Okay, now we're back where we should be. We're starting with our template, which is always a good place to start. So our template, if we copied it, if we're doing DNA replication and we need to copy that template to make our complementary strand of DNA, that A would be a T. That G would go to a C. A would go to a T. C would go to a G. T would go to an A. And that C would go to a G. And then we would also go back to that template to make our messenger RNA. So our mRNA, the A would go to, not a T this time because it's RNA, it's going to go to a U. A will go to U. G still goes to C. A goes to U. C goes to G. T still goes to A. And C still goes to G. And that's what that one will look like. And the next one we're given the complementary DNA, so we need to go back to our template, make our template here. We're just kind of working backwards. G would have been a C. A would have been a T. T would have been an A. A was a T. A was a T. And G was a C. Now we can use our template to make our messenger RNA. That C would go to a G. That T would go to an A. A would go to a U because it's RNA this time. T would go to an A. T would go to an A. And C would go to a G. This is what the polymerase will be doing, laying down these complementary bases. And now we're going to take our messenger RNA. We've got to go backwards to find out what our template was. So if it coded for a C, it was a G. If it coded for a U, it was an A. G was a C. G was a C. C was a G. U was an A. A was a T. C was a G. And A was a T. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this mRNA would code for 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 3 triplet codons in there. So that sequence would code for 3 amino acids. There's 3 groups of 3 in there. And now we can do DNA replication. We're going to go from our template to our complementary DNA strand. This G right here would be a C. A would be a T, because we're talking about DNA this time. C would be a G. C would be a G. G would be a C. A would be a T. T would be an A. G would be a C. T would be an A. So what's that one? Okay, we'll answer a few questions. We won't get to all of them. So we're going to mRNA. So T would be an A. C would be a G. T would be an A. G would be a C. A would be a U. And G would be a C. What is copied during DNA replication? And that would be a DNA template. It's a strand of DNA. How many strands of DNA are used to make complementary strands of DNA? So you're going to be copying both strands of DNA when you're doing DNA replication. And to make 2 complete strands, one would be one side's the old and one side's the new. So intron is cut out